So it's my great pleasure to welcome Dr. Jose Eduardo Meireles, Dudu for short, to join us tonight. Dudu first attended the Rio de Janeiro State University, where he earned his BS in biology. He then received his Master of Science in Plant Biology at the Campinas State University, also in Brazil. While there in Brazil, Dudu worked as a research fellow at the Rio de Janeiro Botanical Gardens, working on the systematics of the Fabaceae. He holds a PhD in biology from Duke University, where his dissertation focused on evolutionary ecology of plants. He completed his postdoctoral work at the University of Minnesota, focusing on spectral biology. Presently, we're so lucky to have Dudu in our region. He's assistant professor of plant evolution and systematics and director of the University of Maine Herbarium in Orono. And on a personal note, I'm particularly grateful to Dudu because he, along with our NEBC curator, Lisa Stanley, is serving as a member of the Plant Science and Collections Advisory Group at Coastal Maine Botanical Gardens. So thank you for that, Dudu. His lecture tonight will be Plant Diversity Shows Its True Colors. Thank you so much for being with us tonight. Please join me in welcoming Dudu.
Thank you, Melissa. That was much too kind. And yeah, I am honored to be here and for the opportunity to share a little bit about the research that we do in my lab. So first of all, thank the University of Maine, the Ascend Institute, which is an NSF-funded institute for spectral biology that got funded, and for NSF. So Earth is home to an incredible diversity of plants, right? And you know that that diversity of plants has profound impacts on how ecosystems work and for how planet Earth functions, carbon cycle and whatnot. And for a very long time, we've known now that diversity is more than the counting of species, right? It encompasses their evolutionary history, so where they fit in the tree of life, the amount of genetic diversity that they have, and also the variability in how they function, all the ranges of tolerances and ecophysiology and attributes that different plants have, and that allow them to inhabit different places. It's always mind-boggling to me that, you know, this little duckweed and this giant sequoia like shared an ancestor not very long ago. So it'd be a very foolish thing to, you know, try to understand the diversity of plants by counting species and not accounting for the differences between those two guys. Unfortunately, we also know that this diversity that we rely on is facing, you know, really important challenges. So, you know, climate change, land use change, etc. All of those are really changing this diversity that we rely on. So it is critical that we figure out ways of assessing that diversity. Again, not only the counting of species, but all those other dimensions of diversity too. And unfortunately, we do not have a magic wand, and I can't make up, you know, 100 million of you and ship you all over the place to assess diversity of plants. We have to figure out some other mechanism, some other way of assessing that diversity. So that is the central question here: is how do we assess the evolutionary and functional diversity of plants at scale? In other words, as we try to measure that over many different regions of the world and over time. And my answer to that is using light. So pretend that we're trying to figure out whether we should eat a tomato, okay? Now, I love my dog. He's really cute, but he's got a problem. He can only see one color. So this is the way my dog, Butter, sees the world. Kind of boring. It's hard to decide from that whether you're eating that tomato or not. Turns out I also love my dad, who's colorblind, and he sees two colors, and that is the way he sees the world. But this is the way my daughter, who's not colorblind, sees the world. There's a lot of variation, a lot of information that you can extract from the world that you can take in. If you can see a third color. Let's think about this. You're looking at a photo and you're extracting chemical information. You're inferring, how much sugar is in those tomatoes. If they're like tannins that you don't want to eat or whatever. And we can do all of that using 3 colors. Well, imagine then if you can see thousands of colors, and not only in the visible, so from the blue to the red, but across a wider range of light. So this is a little part of the electromagnetic spectrum, going from X-ray to microwave. In this little sliver, here's a visible spectrum, what we can see. There's much more there. So what we do when we talk about measuring the spectra or measuring colors in plants is actually taking this piece of equipment. Second, we're going to say, okay, telling an oak from a pine using color, that is easy. Throw a hard problem at me. We're not doing sedges yet, but those are too hard. We've been looking at really closely related plants that hybridize and biological problems like that. Third, we're going to say, okay, can we move that from live plants and try to leverage herbarium specimens to learn about the function of these plants? Can I go to a herbarium and try to learn, if nitrogen content in plants have been changing over time in a non-destructive way? That is a big part of this. Takes 6 seconds, you're not damaging anything, you're not taking any tissue from a herbarium specimen. So there are challenges there, and we're going to talk over those a little bit. And finally, if we have time for it, we're going to talk about the evolutionary aspect of it. In other words, how do we reconcile these spectral measurements, the measurement of light of leaves, with the theory of evolution and how attributes of plants change over time. Cool? So on to the first bit. The goal, as I said, of this project was to try to map out the variability in the spectral measurements across a whole plant drift life. And we're going to talk about 2 specific questions. The first one is, can we identify plant lineages using that method? In other words, can I bring my instrument over here, measure a plant without looking at it, or without, not asking anyone, and tell you what that is just from the fingerprint of that signature of light. And the second is, if that is the case, can we figure out where in that signal the information about evolution is? In other words, is the visible part of that spectrum where the pigments are and whatnot, is that what's telling me what different plants are? Or is it, in the longer wavelengths where secondary compounds and things like that are? So if you're getting hints of like chemotaxonomy, there was a thing a while ago. There are synergies here. So for this first project, what we did was measured 16,000 leaves for about 550 species. Then we did, phylogenetic reconstruction using DNA. So we recovered a big phylogeny, and that map there shows you, the distribution of the plants we worked on. And lastly, the thing we're doing, and we don't have to worry too much about the details, is us use that metric called phylogenetic signal to assess where on the spectral curve the information about evolution or about relatedness is. And all we have to that metric's essentially, how much, how does how does the evolutionary, to what degree does a phylogenetic tree help me predict something about the plants? Okay, so think about like succulents, and if I tell you that something is in the cactaceae, you're gonna say, yeah, that's probably succulent, right? So we don't have to pour over the details, but that's the idea. Like, how can I tell you where something is on the tree of life, and how much does that inform you, about the characteristics that a plant will have? So that is phylogenetic signal. A high number means evolution tells you a lot about what a plant looks like, and a low signal means not a whole lot. So now we're going to answer our first question. Can we tell lineages apart using these color measurements? And this is, it's not a confusing figure, but let me walk you through. So we did this using machine learning. The methods really don't matter a whole lot. But what you're looking at is a matrix called a confusion matrix. And I was confused by that for a long time. So the bottom line here is the diagonal on this square, the entries on the diagonal, that's where the correct classifications were. In other words, a plant was an asterid, and the light measurement told me, oh yeah, that is an asterid. If it's in some other entry, it means that the model predicted it wrong. Does that make sense? So all we have to care about here is like how much color we have on the diagonals versus the off diagonals, right? And as it turns out, these light measurements do pretty well. So you can tell, broad lineages of plants apart. So asterids from gymnosperms, from monocots, using those measurements. Well, you can go down to different orders of plants or different families of plants and still do a pretty good job. So all of those diagonals are filled with solid colors, right? Or most of them. So that means that you can give me can take a piece of leaf that has no other information. You're not looking at it. You're doing a scan of the color, and you can tell to what family that thing belongs to. And this slide, real quick, just tackles the second point. It's like, okay, if light can tell you about the identity of a certain plant, where does that information live on this spectrum right here? So this plot on the top is that phylogenetic signal thing that I told you about. So how important is phylogeny for predicting the how important are different colors for predicting the identity of a plant, right? Or its phylogenetic placement. And the bottom line here is the answer is different for different lineages. So if you take gymnosperms, most of the information about how different gymnosperms are related to each other come from this region of the spectrum. That is this line over here in brown, come from this region of the light measurement. Now, if you go to magnolias, a lot of that information is also coming from this other region. So there's no silver bullet, essentially. And all of you are botanists, this is not surprising, right? If you want to ID an oak, you're going to look at different characteristics than, if you want to ID a magnolia or something else, right? So there's no silver bullet characteristic that is telling you what a plant is. The same is true for these light measurements. So our big conclusion is that the evolutionary history of plants is captured by light. We can use those measurements to identify plants and essentially tell where they are on the evolutionary tree. This is a time where we actually throw a curveball at this thing. It's like, telling an oak from a pine, that is very easy. My 4-year-old can do it. And she's not a botanist quite yet. But the idea here is like, if we throw a hard problem at it, can it deal with that? So this is work by a grad student of mine and by a postdoc who's now a postdoc here at Harvard with Chuck and has been doing a lot of that work now, Dawson White. So the question is, can we detect the diversity in like the fine scale diversity in plants using those same methods? And here's an example. Here, this is Dryas, and those are 3 species of Dryas, and they're kind of hard to tell apart and messy. If you go and look at the biology of these things, or the evolution of these things, there are 2 problems. One is that they have a really recent diversification, so they've been speciating quite recently. There hasn't been a lot of time for big differences to accumulate between them, the different species. And the other problem is that they hybridize. So there is genetic communication between species. And again, that is the core question. Can these spectra capture diversity when the biology is complicated and messy? So everyone knows about Dryas. It's important geology, as a matter of fact. Important alpine plant. And here in North America, so we did this work in Alaska. That's just like the circumboreal distribution of all of Dryas. But we did this work in Alaska, where there are 2 species now, Dryas alaskensis and ionensis. And Dawson was the person actually doing a lot of the genetics and then starting looking at the taxonomy of this. So what we did here is we went to Alaska and collected a bunch of Dryas in different mountain ranges. They're all close to each other, and most mountain ranges actually had the 2 species together. They're intermingled. And every once in a while, you'd see something morphologically kind of looks like a hybrid. So you would tag as a hybrid and collect it too. So that went there, made herbarium specimens, we collected the tissue for DNA work, and we measured light in those guys. There's some genetic work, and all that this, that top plot is a PCA analysis, so a clustering analysis of genetics. And all that we really care here about is that we have one species, Dryas elascensis, to one side, Dryas agenensis to the other side, and the hybrids in the middle, genetically, right? That makes sense. This is a similar kind of analysis. It's a clustering analysis, but showing, different populations of Dryas agenensis here, the hybrid right in the middle, and Dryas elascensis on the other end. But it's just to show that they're two separate, the two species are genetically disconnected, they're genetically dissimilar, but they do hybridize. And there is variation in between populations. So it's not like every Dryas, ionensis and Alaska is the same, or in that mountain range the same. There's variability. The second interesting thing that kind of happened with the hybrids is we went ahead and did a lot of kind of not necessarily morphometric measurements, but a lot of morphological measurements on these guys. And this is a little matrix that is showing the correlation between different traits on their leaves. So if you're thinking about the two parent species, and let's say we look at how hairs are correlated with scales on the leaves. Over here, you see that they're really strongly positively correlated, right? So if you have lots of hairs, you tend to have more scales. Does that make sense? So there is a correlation between these traits. So it means that you can cluster the two parents morphologically. But when you go and measure the hybrids, that starts to fall apart. Now you can have a hybrid, one of the hybrids has lots of hairs, but not many scales. So it doesn't fit the pattern. Does that make sense? It's just more variable morphologically. And the whole idea was, okay, this, this is a challenge if you're doing, taxonomy, we do based on morphology. And we kind of predicted that it was going to be a real hard problem for this light technique too. So we went and did the whole machine learning thing again. And as it turns out, these are the confusion matrices that we looked at. We can predict the different species very well just from the light measurements, nothing else. So those are the two squares over there, and the hybrids to 75% accuracy. If you look at the population structure, so can you tell me from what mountain range this Dryas elascensis comes from? You can do the light measurement or take that light measurement and tell the population. We went ahead and we went a step further, and this is these are metrics of genomic differences in the genome. Okay, so the amount of ancestry in the genome that is one species or another. On the x-axis, you see the reality, and on the y-axis, you see the genomic differentiation as predicted just from the light measurement. And there is some variation and whatnot, but it's I did not want to believe this. I want to keep my job as a taxonomist. So it's I have mixed feelings about these things. But, the conclusion that we took from this is that this fine scale diversity in plants, won't necessarily be overlooked as we try to use these, spectral based techniques or these light based techniques to assess diversity in the field. I'm not saying that this is going to be as good as having the expert botanist look at every plant. But, as a first cut type of assessment, I think there's a a compelling case here. Now, as Melissa suggested, I'm the director of the herbarium in Maine. I love herbaria. I, could live in one if I could. And it's always nagging. So can we leverage herbarium specimens to do this again, to try to learn about the function of these plants? Can we use these techniques to try to identify things that have been unidentified forever in a herbarium, or at least give us a couple options? So can we put these, it's two questions. It's like, can we make efficient use of herbaria? And then can we give back? Can we, take these things that we're using in remote sensing and whatnot and actually, leverage them in the herbarium, use it as an additional tool? So I don't have to preach to the choir here. Herbaria are the ultimate plant database, right? Tons of species, everything identified. So you may recognize a few of these folks. Garth is over there merrily. Yeah. So there are tons of plants in herbaria that can leverage to do these measurements. And the second really important aspect, and I think that gets lost to people that are just doing genetic work or people that are just measuring traits. And that is, if you don't have a herbarium voucher, your data just isn't as valuable. Taxonomy changes, species get split. And if you have this huge data set that is attached to a name, and that name changed, or there are two things now from that name, what do you do with your data? So it is, my view, it is critical that whatever is happening, if it's, molecular work, if it's these, DNA or this like spectral based work, that all of that is integrated with herbaria. Though they're the ultimate, database or the ultimate source of knowledge about plant diversity. So there are challenges to doing this in herbaria, right? As it turns out, it's a plus and a minus. Several of these specimens are old, like real old. They're mounted on paper, so they have a background that is not standard. So there are a few things that we want to, technical things we want to tackle. First is, does the paper background, or there's whatever background you have in your herbaria, does that really affect things, your light measurements? If you're planning on scaling this up and, coming to the Harvard herbarium, measuring everything, would it even work? Or how would that paper affect things? Leaves age in the herbarium. They're not like that, that color when you collected them, or, a week after you dry them out. So how does that aging process affect what you can tell about the function of a plant, the amount of nitrogen it had, or the amount of chlorophyll it had, or does it change your ability to tell species apart if they dry out and senesce? And we're expecting for those two things to be the case. So the third question is, can we actually take an old specimen and using the computer make it new? In other words, can we take the light measurements on an old specimen and say, oh, what would it look like were this specimen new? Can we artificially rejuvenate them? And that is all with the goal to assess the identity of plants or their function using these light measurements. So here's what we did. We went out on campus in Orono and collected a bunch of fresh specimens, red maples, different oaks and whatnot. And we measured them all fresh, and we measured their chemistry and everything. Then we pressed them and put them in a herbarium, and we've been letting them age. And every once in a while, we go there and measure the light. And we measure that under kind of three different conditions. One's the loose leaf, as if it were in a herbarium packet. The second is just against the paper background. Imagine like something that's just taped. And third is, slap some glue in there and see how much that changes things too. And so here are different of these, a bunch of these different curves, and we don't have to pay attention about the details or what these plants are. That's a red maple, Quercus alba, cattail, but whatever. The key point here is, in red, you see the leaf that is measured, is a loose leaf. So it's measured against your regular black background in a standard way. In green, you have a specimen that is mounted and glued to the base, to the mounting paper. And in blue, you have something that's just like it's been taped, but not glued. And the bottom line here is, the background in gluing versus not gluing actually changes your measurements, which is problematic, right? So we'd have to figure out a way of dealing with that. Of course, this actually varies with characteristics of the leaf. So as you can imagine, a thin leaf here to your left is more affected by that background effect than a thick leaf, right? A thick leaf, like with a thick leaf, you don't necessarily see a whole lot of the background. Most of the light is coming from the actual leaf. But if it's paper thin, a lot of that background shows through. And the second problem, so background matters. The second problem is the age also changes those signals pretty significantly. So in red now, you see a fresh measurement. And, 2, 3, and 4 are just 4 separate steps after they've been dried. So I can't remember exactly. It had been 2 weeks, 4 weeks, and then between 3 and 4, it had been 2 months or something like that. But you can see those leaves aging in the herbarium and their light signature changing over time. And we don't have necessarily to pour over this thing. This starts to get technical. But let me hit this one. The bottom line is we can actually revert that process using modeling. So the top left panel shows a bunch of these light measurements that are old, okay, in things that have been aging in the herbarium for 2 months. That's in black right there. But remember, we also measure those same plants when they were fresh, right? So we can build a model and actually rejuvenate them. So the bottom left plot here shows in the orange curves are the black curves after they were transformed by that model. They were rejuvenated by that model. Does that make sense? And the blue is just the truth. So the bottom line is that you can get something that is pretty close to what those curves looked like when the samples were fresh. Of course, it's not perfect by any stretch of the imagination, but it is way closer to the blue than those black curves were, right? And as it turns out, if you try to identify different species using that technique, you can do a very good job, essentially a perfect job with the fresh leaves. But you can do also an amazing job if you're using old herbarium or like older herbarium specimens. That's the stuff on the right. So it was actually 4 months. So it may not really matter that these plants are changing over time with age. You can still tell them apart and learn from herbarium specimens using those techniques. So that is a conclusion of this. It's we can actually learn things, useful things using these spectral measurements from herbarium specimens. But we'll probably have to come up with, come over here and go around the country, Florida and Field Museum and whatnot, and establish standard protocols on how to measure these things, how to minimize all of those artifacts. I think my overarching conclusion is that these measurements of light are incredibly powerful. They're not going to replace molecular work. They're not going to replace the work of a botanist by any stretch of the imagination. But as we think about the challenge of assessing the diversity of plants, in New England alone, it is a hard problem, right? And this is kind of like a best case scenario. Think about the Atlantic rainforest where I'm from, or places in the Amazon. We really need to devise new techniques, new technologies to try to understand which plants are around us and how they behave in the world, why they are where they are, and things like that. So I think that's my overarching conclusion, that we should seriously consider these techniques or these models and keep that on our radar, because it's I think it's coming. It's not long from now, we're probably going to be scanning a ton of herbarium specimens using these things. We're already flying these equipments on airplanes and mapping species and stuff like that. So it is it's a super powerful technique, and I think there's a lot of synergies between field botany, citizen science, herbaria, and these advanced techniques. So that's all I had for you guys today, and I'm happy to take any questions. Thank you.
One of the early slides had a image of a device to get clamped on a leaf to do the photometry on the leaf. How much does that thing cost?
I know, because I have one in the lab. It's like $80,000. Yeah. So the good news is that you know you so those are like the really fancy ones, and the way the way you can think about this is like we get these high power machines that measure things at an incredibly fine scale, but you don't necessarily need all that fine scale. So once you start learning, you know, the about the exact things you want to measure and whatnot, you can design instruments that will target what you want. That already happens in agriculture. If you take a little instrument to measure chlorophyll in the field, or plant stress, or water, like all of those are based on light. They're based on these techniques. But instead of measuring 1,000 bends and being cooled and this and that, you're simpler and cheaper. Yeah.
So how is this being used to really assess plant biodiversity on a small scale or on a state scale?
It's been used in like various different ways. So at the smaller scale, what we typically do is you go out in the field and you collect your plants, and if you're thinking about that broader definition of diversity that encompasses functional diversity, for instance, all the different characteristics, you end up phenotyping these things. So you take these measurements, and along with the identity of the plant that you have and their location, you're assessing nitrogen, you're assessing phenolics, you're assessing this and that. That all happens at the leaf level scale. But we've been flying equipment like this in on UAVs, and have a proposal now to put one of those on the university airplane. So you can cover a lot of forest using this. There's a researcher, Greg Asner, who was at Stanford and moved to Arizona now. But he's been doing this in the tropics, not necessarily using the evolutionary approach or anything like that, but has been super successful at detecting different species, or just in general, how much. It's fun. It's like how much variability you have. How many different things it looks like you have in a certain spot, even though you don't know what the plants are. That they're it seems to have a lot instead of few. And he did a lot of work in Peru. They actually established a national park based on off of that work. That work identified a few spots that were had a lot of like plant species diversity and functional diversity that wasn't covered by any other conservation area. From that work, botanists were hired. People went there, did the field work, and like did the field part of that, and established a new park. So so that's happening in the tropics. That's happening here in the US. So it varies from scale from leaves all the way to airplanes. But yeah.
Very interesting talk, information. Thank you. I have a comment. Since in Australia there are more than 900 species of acacia, and at least about 700 species of eucalyptus, it will be interesting if someone does this kind of work. And just that's a comment. My question: does the spectral analysis does it differentiate between C3 and C4 leaves, and also leaf-like branches? For example, in asparagus and custard, they are not leaves. Does it distinguish between the real leaves and these modified branches?
Yeah, I mean, so you can definitely distinguish through C3 and C4s. That is not not problematic, and I've seen that work. Regarding like, let's say photosynthetic tissue that is not a leaf necessarily, so stem, or think like a modified like petiole, whatever that is. So you can do the same kinds of thing. You don't necessarily have to measure a leaf, and I'm willing to bet that you can tell that it's a different structure because of the thickness and whatnot. But I don't know if anyone has actually looked at it. Your comment about these super diverse clades, that is 100% true. I mean, I have a hard time thinking that this would work very well on a crazy large genus like lupins or anything that's super diverse, very recent radiations, and lots of species. So I think it's going to be mostly broad strokes. And if you're targeting a large species complex, the way I would think you could use this is essentially as another dimension of morphometrics or chemometrics. It's another set of data that will allow you to find differences in clusterings between species, but it's probably not going to be your only answer.
So to clarify, whether the species by species in the model or the
Sorry, can you repeat that question for the Zoom people?
Sure. Let me just make sure that I understood it. So, is a question that do we need to measure every species that we'll want to identify?
Yeah.
Yeah, that is a great question. I didn't quite get. Maybe no, we're not going to have the time to do that. But so that is a problem in machine learning, like how you can only detect things that you already know about. So that is a problem with supervised learning. And I mean, if you don't stop me, I'll actually do this. So
You're not sharing on Zoom anymore.
oh, I'm not?
No, I took you off.
Oh, can I reshare? Okay. So hopefully this is going to work. But here's there's a whole part of this stuff that's like, there are things you already know. Yeah, great. You can tell them apart. What about the things that you don't know? How do you do that for things that we don't know? And there is an answer that is very similar. I mean, it's kind of inspired by my background. That's like DNA barcoding. So essentially, we have part of the tree of life that has a lot of these measurements for, right? And we know that tree. Someone recovered that. But then, you measure something that's unknown. And the question is not anymore, is it this or that, but where does that unknown land on that known tree of life, right? It's kind of like your barcoding. So you take a new measurement, you say, does the data support that kind of relationship or some other relationship? And and the whole system is very it uses the same essentially the same math as that's under that's in phylogenetics. But here's the gist of it. This is a modeling exercise of phylogeny that we simulated. We then, took the species out, and then we use that model, that, spectral barcoding, to try to predict where it lands. And it does an okay job. And we've tried that with an oak data set, and it actually kind of works too. So there are alternatives to what I presented. It's just more nuanced and messier. But that's a great question. Yeah.
I have a chat question. Find it. From Julie Richburg. Do you see a future to use this technology to locate individual species, whether rare or invasive?
Yeah, so people have been doing that for an invasive species in different places in like California and whatnot. There's been a lot with aquatic invasive species, Keith, and yeah, so that's already happening. Regarding the rare ones, I don't know for sure. Someone must have been trying, but it is a harder problem, just because you have to locate them in a ton of pixels, and yeah, it's harder. But for invasives, for sure.
What? That looks like there's some similarity index.
The table, it seems like a similarity matrix, but it's really just that the computer kind of like trying to guess based on your model whether it's this or that, and counting how many times you're right or wrong.
It really breaks up the field.
Yeah, it does it families, it does it order, so it does the genera, so you can essentially imagine like cutting diversity at different like taxonomic ranks, or taking a phylogenetic tree and slicing it at different depths and doing the same things. Yeah.
We have one more follow-up question on Zoom to the previous one asked.
Yeah.
Are you looking for invasives down from plains? I think that was not quite clear.
Yeah, so I'm not doing that personally, but there are researchers doing that
With the spectral.
with yeah, it's a camera, just like your regular camera, but measures a ton of different colors instead of your red, green, and blue. Yeah.
One last one from the room. It's really hard to track the effects of climate change on biodiversity. I'm curious if you think this could evolve into a score or something that people can use to track the effects of.
That's a great question. I think there are two parts. I think one of them is, how do you try to summarize like these things into something that's like a score that's easy, to understand. Like think about like the stock market. You don't have to know everything. You just have to know if the S&P went up or down, or like something like that, right? So I think that is an important part of biodiversity science and conservation science is work on that. The other side is for sure. So NASA got approved. The Academy of Sciences actually mandated them to develop an instrument like the one I've shown that's gonna live on a satellite. The mission's called SBG. So it's Surface Biology and Geology, and the goal is to map the geology and the biodiversity of the planet. So, they've been developing the instrument. It should be, hopefully out in like 2027, or these things delay. But but folks have been eyeing that kind of thing. How to do these diversity assessments, and especially as climate change hits, at a truly at a global scale.
Well, this has been absolutely fascinating, Dudu. Thank you so much for coming all the way down to
Oh, thanks. I
Cambridge
mean,
to
it was a
deliver the
pleasure.
lecture for
Yeah.
us, and we just so appreciate your coming.
Yeah, and sorry that I didn't have a lot of plants. It's like I always feel bad given these talks. Like I need more plants there. Yeah.
It's perfect. Thank you so much.
Thank you.